Hello everyone and welcome. Today I'm excited to introduce you to our presentation on the T4 phage. This is the first slide, so I'll keep it brief. We'll be exploring what T4 phage is, its structure, and its significance in microbiology. I hope you find this session informative and engaging. Let's get started. Welcome to the introduction slide. Here, I want to set the stage for our entire presentation by highlighting some key points. First, we'll explore the main ideas that underpin our topic, giving you a solid foundation to understand the details that follow. I'll explain why this subject is important and how it connects to the broader themes we're discussing. The goal is to give you a clear overview and spark your interest in the specifics that will come later. Remember, this is all about framing our conversation so you can see the bigger picture and understand how each piece fits together. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a closer look at the T4 phage. This is a classic example of a bacteriophage, which is a virus that infects bacteria. The T4 phage is particularly well studied because of its complex structure and its role in understanding viral replication. If we think about its shape, it has a head, or capsid, that contains the genetic material, and a tail that it uses to attach to and inject its DNA into a bacterial cell. This mechanism is quite efficient and highlights the sophisticated ways viruses have evolved to infect host cells. The T4 phage is also important in research because it has served as a model to study molecular biology concepts like DNA replication and gene expression. Connecting this to our overall theme, understanding phages like T4 helps us appreciate the diversity of viral strategies and their potential applications in biotechnology and medicine. Let's take a closer look at the structural components of this virus, starting with the head, also known as the capsid. This part has an icosahedral shape, which means it's a 20-sided figure, pretty efficient for enclosing the viral genetic material. The capsid is made up of protein subunits, which come together to form this protective shell. Inside, you'll find the viral DNA, which is linear and double-stranded, known as dsDNA, and it's about 169,000 base pairs long. The main role of the head is to protect this DNA from damage and ensure it remains intact until the virus infects a host cell. Moving on to the tail, which is quite fascinating. It's long, rigid, and contractile, meaning it can shorten and extend, which is crucial during infection. The tail consists of two main parts, the sheath and the core. The sheath acts like a syringe, helping the virus inject its DNA directly into the host cell. This structure is essential for the virus to successfully deliver its genetic material into the host, kickstarting the infection process. Connecting these features to our overall theme, understanding these structural components helps us grasp how the virus is designed for infection. The head protects and stores the DNA, while the tail acts as the delivery system, both perfectly adapted for infecting bacteria, which is what makes these viruses so effective. Now, let's take a closer look at the structure of the bacteriophage, starting with the base plate. This is a hexagonal structure located at the tail end of the virus. It plays a crucial role because it contains receptor-binding proteins, which are essential for the virus to recognize and attach to its host cell. Without this part, the phage wouldn't be able to initiate infection properly. Moving on to the tail fibers, these are long, hair-like structures attached to the base plate. Think of them as the virus's antennae. They help the phage recognize and latch onto specific receptors on the surface of E. coli bacteria. This specificity is vital for ensuring the virus infects the correct host. Next, we have the tail sheath. This is a protective covering that surrounds the tail tube. During infection, the tail sheath contracts, kind of like a spring loading, which then helps inject the viral DNA into the bacterial cytoplasm. This contraction is a key step in the infection process, enabling the virus to deliver its genetic material efficiently. Finally, the tail tube itself is a hollow structure inside the tail. The viral DNA travels through this tube as it makes its way into the host cell. It's essentially the delivery pathway that ensures the genetic material reaches the right place inside the bacteria. Altogether, these components, base plate, tail fibers, tail sheath, and tail tube, work in concert to recognize, attach, and inject viral DNA into the host. Understanding this intricate structure helps us appreciate how the phage infects bacteria so precisely and effectively. Now, let's take a closer look at the properties of T4 bacteriophage. This virus is a classic example in microbiology and provides a lot of insight into viral structure and function. First, T4 is a double-stranded DNA virus, which means its genetic material is DNA, similar to our own cells, but it's packed inside this complex protein coat. Speaking of which, the T4 phage has an icosahedral head that protects its DNA and a tail structure that it uses to inject genetic material into bacteria. This tail is quite sophisticated, with tail fibers that help the phage recognize and attach to specific bacterial receptors. Once attached, the tail contracts, injecting the DNA into the host cell, which then hijacks the bacterial machinery to produce more phages. T4 particles are relatively large for viruses, about 200 nanometers in length, making them easily visible under an electron microscope. These properties are important because they influence how T4 infects bacteria and how we can study or even manipulate such viruses in research and biotechnology. Connecting this back to our overall theme, understanding these properties helps us grasp the mechanisms of viral infection and the potential for phage therapy as an alternative to antibiotics. So let's dive into how T4 phage infects bacteria through its lytic cycle. Once it encounters a susceptible E. coli cell, the process begins with adsorption. This is where the tail fibers of the phage recognize and attach to specific receptors on the bacterial surface. Think of it as a lock and key mechanism that ensures the phage binds only to its target. Next comes penetration. The tail's sheath contracts, acting like a hypodermic needle, injecting the viral DNA directly into the bacterial cell. This step is crucial because it delivers the genetic material needed to hijack the host's machinery. Once inside, the phage DNA takes over the bacterial cell's processes. This is the replication phase, where the phage DNA directs the cell to produce new phage proteins and copies of its DNA. Essentially, the bacteria become factories for producing more phages. After enough new viral components are assembled, the process moves into assembly. Here, the newly made phage particles are put together inside the host cell, ready to be released. Finally, we have lysis. The bacterial cell bursts open, releasing a large number of new phages, typically between 100 and 300. These new phages can then go on to infect neighboring bacteria, continuing the cycle. This entire process exemplifies how the T4 phage efficiently propagates by rapidly killing its host and producing many progeny, which ties back to the overall theme of viral infection strategies. Let's start with host specificity. This phage infects Escherichia coli specifically, meaning it has a very targeted approach. It recognizes its host by using tail fibers that bind to specific receptors on the bacterial surface. This precision is crucial because it determines which bacteria the phage can infect. Moving on to its life cycle, this phage follows an obligate lytic cycle. That means once it infects a bacterial cell, it hijacks the cell's machinery to produce new phages, and then causes the host cell to lyse, or break apart, releasing the new phages to infect neighboring bacteria. This rapid destruction of the host cell is a key feature of its strategy. Now, looking at applications in biotechnology, this phage is incredibly useful. It's employed in genetic engineering and molecular biology studies because of its ability to target specific bacteria. It's also a promising tool in phage therapy, offering an alternative to antibiotics, especially against resistant bacteria. Additionally, it's used in bacterial detection methods, particularly in food safety, to identify contamination quickly and accurately. All these points highlight how understanding the phage's host specificity, life cycle, and applications connects to the broader theme of harnessing viruses for scientific and medical advancements. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide.